Swayam Prabha. Digital India. Educated India.
In previous lecture, we have discussed about the electrophoresis of proteins. We have discussed about various techniques used in the analysis of protein molecules or polypeptides which may be smaller. If you could recall, we have talked about STS page electrophoresis, which is a form of discontinuous gel and developed by lamellae. We have talked about isoelectric focusing, where pi value is taken for separation, and proteins in the electrophoretic gel, they focus on a particular pH which equals to the pi of a protein or a amphoteric molecule. Also, we have discussed about 2D gel electrophoresis. The most widely used one is which combines isoelectric focusing and STS page, and there the whole proteome could be analyzed, and two particular systems which could be a native and deceased, or say if you are looking for overexpressed protein, could be analyzed. Now, in today's lecture, in this lecture we are going to discuss about electrophoresis of nucleic acids, and here we are going to discuss various techniques which are used for in the analysis of nucleic acids. Now, nucleic acids means DNA or RNA, and mostly it is the DNA which is utilized. Now, nucleic acids could be separated by both polyacrylamide gel electrophoresis and agarose gel electrophoresis. Now, agarose gel electrophoresis is a commonly used method to separate nucleic acids, and polyacrylamide gel, as we have discussed in previous lectures, that polyacrylamide gel is utilized for the smaller fragments of DNA, or small sized DNA is analyzed on polyacrylamide gels. So, the agarose gel electrophoresis is the most common method used. In agarose gel electrophoresis, nucleic acids, that is DNA or RNA, both could be analyzed. This we have also discussed in the horizontal gel electrophoresis. So, the negatively charged DNA molecule or the phosphate backbone of the nucleic acids gives them a uniform charge to mass ratio. Therefore, migration of DNA or RNA in gels is inversely proportional to the size of the nucleic acid. The structure of the nucleic acid, like for example, it could be linear, it could be circular, double stranded, single stranded. So, all these structures of the nucleic acids will also affect their migration in the gels. Now, it is possible to distinguish various DNA fragments that differ by as little as 1%, and in some applications, say for example, in DNA sequencing where it is mostly done on polyacrylamide gels, fragments that differ in size by a single nucleotide can be resolved. So, a wide range of sizes could be analyzed on this gel electrophoresis. It could range from less than 10 base pairs to 20 over the 20 kilo base pairs. Agarose is the most common matrix, as we have discussed earlier, for the electrophoresis of nucleic acids, and this is mostly done in horizontal electrophoresis setup. So, nucleic acids ranging in size from say 100 base pairs to 20 kilo base pairs can be resolved depending on the agarose concentration, if you could recall when we were discussing horizontal gel electrophoresis, you can go from 0.7% to say 3% agarose can be taken. Now, acrylamide, which we will discuss in a little bit in DNA sequencing gels, is used for only smaller sized nucleic acids and oligonucleotides. Now, if you see in this table, this gives you the percentage of agarose or acrylamide and the fragments that could be separated. If you could pay attention here, then 0.7 agarose can 0.7% agarose gel can fractionate or separate say from 0.8 to 20 kb. Likewise, up to 2% agarose can give you 0.1 kb to 3 kb separation. So, depending on like as you increase the concentration of agarose, the pore size will decrease, and so the size of the DNA which is separated will also be smaller as compared to one with 0.7%, where the pore size will be much larger. Now, in acrylamide, where pore size is much smaller as compared to agarose gels, you can see 3.5% acrylamide could be used for only say very small like 100 to 1000 base pairs only. And likewise, when you increase the percentage of acrylamide say to 5% and up to say 12 or 20%, you can see the size of the oligonucleotides which can be separated also becomes smaller. Like say at 5% it is 80 to 500 base pairs, at 12% it is around 40 to 200 base pairs, and likewise as per the pore size these fragments will be separated. So, the larger the pore size, then larger fragments can be separated, and smaller the pore size, that is higher the concentration of the matrix, then smaller size fragments could be separated. So, the agarose gels are usually poured and electrophoresed in a horizontal apparatus, and this horizontal apparatus provides more support for the gel, and it is easier to prepare. If you could recall, because in vertical fashion these agarose gels most of the time they break or they are not able to be in vertical positions. So, best way to run them is horizontal position. Now, shape as we said also affects the migration of nucleic acids in the gel. So, like structure we have seen, like it could be linear, it could be relaxed, circular, and all those things will affect, as well as shape will also affect. Like for example, RNA and single stranded DNA can form secondary structures due to internal base pairing, and that will certainly affect. So, these secondary structures will lead to a lower mobility. So, different factors will affect the mobility of DNA in the gel. Now, secondary structures or this base pairing could be eliminated by running the gels under denaturing conditions. Like for example, you can use 7 or 8 molar urea, even you can have elevated temperatures for running this gel, and which will prevent the formation of base pairs in RNA or single stranded DNA. This is typical electrophoresis apparatus for horizontal. It is a typical horizontal gel electrophoresis apparatus for running the agarose gels. As you can see that here there is a tank buffer is put, and there will be 2 electrodes, which will be one will be positive and negative. The STS gel is placed in here, the agarose gel is placed in here, and then a sample will be loaded, and this we have discussed earlier also, that wells are created and sample is loaded, and the DNA will run towards the positive electrode. Now, lot of different agents, denaturing agents are utilized, such as urea, formamide or formaldehyde are included in the gel buffers for the electrophoresis of single stranded DNA or RNA, and if size is needed to be accurately determined. So, you have to one has to make sure that there is no internal base pairing which can form secondary structures or double stranded sort of structures which can hamper determination of their sizes accurately. Now, samples are prepared by mixing DNA in a buffer solution that contain sucrose or glycerol, it could be or ficol, Now, this makes the solution dense, as we have discussed in protein gels also. Many times a dense solution in form of glycerol or other things is put in, so that it will sink or it will settle down at the bottom of the well without diffusing, without, like, you do not have to really remove the buffers. You can directly load the samples into the well where buffer is overlaid onto the gel. It could be TAE or TBE buffer. Then certain dyes such as bromophenol blue is also included in the sample. So, it makes easier to see the sample that is being loaded, and also it can be a marker of the electrophoresis front, and same, if you could remember, was used in polyacrylamide gels also. Now, once the system has been run, the DNA in the gel needs to be stained and visualized. So, there could be different fluorescent dyes could be utilized for visualizing DNA, and the most widely used fluorescent dye is ethidium bromide. Now, ethidium bromide is a cyclic planar molecule that binds or intercalates between the stacked base pairs of DNA, and ethidium bromide concentration therefore builds up at the site of the DNA bands, and under UV light the DNA bands fluoresce orange red. So, you can see them very clearly. Now, there are two ways you can stain or you can do the visualization of DNA. One is that ethidium bromide can be copolymerized with the agarose gel. That is, when you are preparing the agarose solution, you can put ethidium bromide in it, and it is in the gel when you have polymerized it. Other method could be that you can run the agarose gel first, and then you can put the gel in the ethidium bromide solution, and so it will be stained after certain period of time. Then the extra ethidium bromide can be washed off with water, and then the gel could be visualized in under UV light. So, both ways it could be done. One important thing here is when you are running, when you put copolymerized ethidium bromide runs in opposite direction. So, many times the bands which run faster on other side of the gel towards anode, they will be hard to see. So, many times you have to add little bit ethidium bromide in the tank buffer on the other electrode. So, it should be noted that ethidium bromide can be harmful also, like if you are doing extensive viewing of the DNA with UV light, that can result in the damage of the DNA by nicking and base pair dimerization. Likewise, like I said, ethidium bromide could be harmful, it is a carcinogen. So, it should be handled with care, and when you have this ethidium bromide stained gels or ethidium bromide solution, it should be properly disposed of after treatment. It should not be directly thrown into the sink. That is a very important in doing these experiments, that you take proper care and you do not harm yourself or others. So, this is a typical structure of ethidium bromide, where you have a cyclic planar structure with bromide ion, and when you load your sample, these are wells where you have loaded samples, and bromophenol blue can be seen, and you can run these samples when they will run towards positively charged electrodes, as they have a negative charge with phosphate backbone. Now, DNA can also exist, like I was saying, there could be different structures. It could exist as a circular molecule, as in plasmids. It could be further more, the circular DNA can exist in multiple conformations, that is, it could be relaxed, it could be supercoiled. Now, if it is supercoiled, it is having additional superhelical twists. When you break a single bond in the phosphate backbone of one strand, it will result in relaxed circular DNA molecule. If you break both strands, then it will result in linear, through, like you can do it through restriction digestion. So, supercoiled circular DNA, if you compare these three, that is supercoiled circular, relaxed circular and linear DNA, and they are of the same molecular weight, they will migrate at different rates through the gel. So, if you compare them, typical order of the migration will be from fastest to slowest will be that supercoiled DNA will run faster, then linear DNA, and then relaxed circular DNA will run slowest. So, what you can see is that the size the structure of the so what you can see is that the structure of the DNA molecule will certainly affect its migration. However, the exact migration is influenced by the agarose concentrations also, the applied current, the ionic strength, and number of superhelical twists. So, a lot of factors to affect, but in general it is supercoiled DNA will run faster than linear DNA and then relaxed circular DNA. Furthermore, it could happen that circular DNA can form dimers or trimers on or concatenated forms, and multiple bands are often seen where you have highly purified plasmid preparation, but you are seeing those because of these dimers and trimers. Now, complete digestion with a restriction endonuclease which only cuts once within the plasmid will result in the formation of single band corresponding to the linear DNA molecule. Now, if you see here in this figure, if these are linear DNA, this is an example of super coiled DNA, this is example of circular relaxed DNA, and these are the what you call concatenated or dimers formation here. If you see the pattern of running, you can see that supercoiled runs faster, the linear, then it is linear, and then slowest is the relaxed one. So, this is the pattern how they will run on a agarose gel. As we have seen, this is how the DNA molecule could be run and separated on agarose gel. Now, DNA could be extracted also from gel. Like if you could recall, we were talking about preparatory gel electrophoresis in the last lecture, and recovery of proteins, as we have seen recovery of proteins, likewise DNA could also be extracted or recovered from the gel. Now, the methods are same. Here what you can do is you can have passive diffusion or electroelution could be done. One way is that a dialysis bag can be placed into an electrophoresis chamber, and the dialysis bag contains this cut out band from agarose. Now, when you apply the current, the DNA fragment will migrate out of the agarose, but will not go out of the dialysis bag. So, the DNA is then recovered from the dialysis bag by precipitation or other methods. So, that way you can drive the DNA fragment out of the agarose gel. Another method involves dissolving the excised agarose gel pieces with say sodium iodide or other chemicals, and then a silica resins which binds DNA is then added to the dissolved gel solution, and following several washes the DNA is eluted from the resin at low ionic strength. Then one can use low melting temperature agarose gels. What happens is when DNA fragment is excised and the gel is melted, the temperatures below 60 degrees Celsius will rarely have an adverse effect on the DNA. And so, low melting gels, melting gels will are very good way to recover the fragments or DNA material, and fragment is then recovered from the solution by ethanol precipitation. So, that could be done. You can melt the gel first and then recover the DNA. So, DNA it is the very commonly or routinely used methods to recover DNA, pure DNA from the agarose gels, particularly from low melting agarose gels. All right. So, this was about the agarose gels or DNA separation on agarose gels. Now, there are a lot of other techniques which are also employed for DNA analysis. One is DNA sequencing gels. Now, DNA sequencing gels have been widely used earlier, and like the final. So, when you do DNA sequencing, there were a lot of processing, a lot of steps to be performed, like one has to restriction digest or DNA molecule to generate fragments. Then those fragments are sequenced, these smaller fragments are sequenced by labeling them, and then by cutting them at specific bases, so that you generate an array of partial array of fragments, which are kind of they are separated by one nucleotide, or they differ in one nucleotide. So, you have an array of fragments, and then those could be run on the electrophoresis gel and analyzed. So, in DNA sequencing method, the final analysis usually involves separating single stranded DNA molecules, which are shorter than about 1000 nucleotides, and they differ in size by only one nucleotide. Now, remember, nowadays these methods may be less used because automated DNA sequencing method and new generation methods have come up, which are much more automated and much more efficient and fluorescence based, where directly data could be collected. But still DNA sequencing gels have been widely used, and it is one method where electrophoresis is used. So, in chemical sequencing method or in enzymatic method, the starting material is a fragment of DNA, and then what is done is the DNA fragment which is obtained after restriction digest, it is labeled at one end, and these end labeled fragment in particularly in chemical method is subjected to 4 separate chemical treatments, wherein each one of these reactions backbone is preferentially broken adjacent to one particular base. So, 4 reactions where 4 types of fragments will be generated, and they are like preferentially breaking the fragment at particular base. So, you have a lot of fragments generated here which terminate at particular base. And now, enzymatic methods like dideoxy methods utilizes polymerase chain reaction to generate different fragments, and where again 4 reactions are there, where you have dideoxy counterpart for each of the base, and a lot of fragments are generated when this particular modified base is incorporated in the sequence during polymerase chain reaction. Now, both methods generate different size fragments, different mostly in single nucleotide. So, to analyze these small fragments, it is necessary to have a small pored gel as compared to that of agarose gels, where the pore size is larger. So, here polyacrylamide gels are used. Here mostly 3.5% to 5% polyacrylamide gels are used to separate DNA in the range of 80 to 1000 nucleotides, and 12% gels, like I have shown you the table, could resolve fragments between 20 to 100 nucleotides. Now, since it is necessary to separate DNA molecules that are very similar in size, DNA sequencing gels tend to be very long, like say from 50 to 100 centimeters. To maximize the separation achieved here, and these gels are much thinner also. They are maybe almost 0.4 millimeter or less thinner, and they are run in TB buffer containing 7 molar urea, and maybe other denaturing agents could also be added, and this is because the single stranded DNA should not form base pair or form secondary structures. And for that reason, they are also run at elevated temperatures, so that these combinations or base pairing is avoided, like they are run at 50 to 70 degree Celsius. Now, once the gels are run, you will find a pattern like this. Now, this is a autoradiograph. So, what is done is, once the gel is run, then the gel will be wrapped in a particular wrapping paper, and then once it is wrapped, then it will be exposed, the X-ray film will be exposed, because it is labeled, these are labeled fragments, and they will expose the X-ray as we have discussed in autoradiography, and then autoradiograph will be generated What you will see is that you will see lot of fragments here, and these are different I said there are different experiments for each base or combination of bases, and they will be read and analyzed, and the whole sequence could be generated. Now, if different fragments are being sequenced, the overlapping sequence can give a complete sequence of a larger DNA molecule. Now, this like after getting the X-ray pattern here or autoradiograph, this is a very tedious job that one has to sit in dark room and then analyze these gel patterns or bands, and even many times artifacts has to be eliminated to get the right sequence. So, DNA sequencing was the very widely used method earlier, but a very tedious method, and it has been replaced by the automated methods, which we are going to discuss in another section. All right. So, if you could recall, as we were discussing about agarose gel electrophoresis, we said that up to 20 kb fragments or 20 the size of DNA of 20 kb can be easily resolved. But these conventional gel electrophoresis system, they have an upper limit that of 20 kb to size that can be resolved, and DNA fragments larger than that are not resolved actually in simple agarose gel electrophoresis. Now, what is the reason, like what could be the reason, like in under best conditions, say if you could go to very low agarose concentration and low ionic strengths, maybe up to 50 kb could be resolved, but still 20 kb is a norm. And what is the reason, like to understand this, there is a process called raptation or snake like movement through the gel. Now, like why 20 kb the fragments or the DNA above 20 kb cannot be resolved. To understand that, we have to little bit understand how DNA runs in the gel. So, let me show you that on your screen a little bit. All right. So, what happens is, like if you could recall when we were talking about the gel, gel is a very random structure, it is not uniform structure. The pores are also like it is a mean, you can say, like they are not uniform sized pores, and there is a meshwork which is a very random structure. So, if I say if this is your gel, if it is a meshwork, if I am making, it is a random structure. Now, what you do, you load like in here you load your sample actually. So, when you one thing has to be remembered that the DNA in the gel is in relaxed form, and once it is put into the agarose gel, it is kind of constrained because of the gel matrix. Now, so up to 20 kb the agarose and the gel interactions are such that the 20 kb fragments could be resolved, but when they reach above 20 kb, then a common phenomenon occurs that we were talking about, and it impedes the motion of the DNA and does not allow the resolving of fragments or DNA molecules above 20 kb. So, how it is, what is happening here is when you apply electric field, then the DNA molecule, it is constrained and it tries to resist the movement, actually it is like to enter to in the pores. So, what happens is that if you have a DNA molecule, if suppose this is your DNA molecule, then one side, which is maybe any side, becomes a leading side actually, it leads here and it will enter. So, one side will enter into the pore, like it will kind of lead and it will enter into the pore. Now, once it enters into the pore, the rest of the fragment, that is lagging side or you can say lagging area of DNA, it will follow that. So, as the electricity is applied, it will start moving, and you can compare this movement to a snake moving in a field which is having lot of bamboo trees arranged in a particular fashion, and it has to move in a I think everybody knows about snake movement. It's a raptation movement. It moves through each of these. It's sort of this kind of movement is occurring. So, likewise same movement occurs of DNA here. When you load DNA and when you apply electric field, there is one side of the DNA leads and enters the pore and the rest of the DNA follows. Now, the molecules above 20 kb, when they move in this DNA, in this agarose gel, after a very short while, as the leading front goes and the rest of the DNA follows it, they are entangled into these you can say meshwork here, and they are entangled so much that their movement is impeded. They are not able to move further. So, even electricity is or electric field is applied, but there is no resolution of the fragments, and this is for DNA molecule above 20 kb. So, you have to switch off the electric field, and again it will relax or reorient, and then it has to move again. So, this problem is the reason why this whole thing that you cannot resolve on normal gel DNA of the size above 20 kb. Now, what is the solution to resolve these large sized DNA molecules? And one solution which is to disrupt the raptation movement. So, pulsed field gel electrophoresis, which we are discussing now, in pulsed field gel electrophoresis, this problem is kind of solved. So, this technique overcomes the problem of resolution of large DNA fragments. What it does is, like I said, you have to disrupt the raptation movement. So, it utilizes two sets of electrodes and that are fixed at angle to each other, and so what happens that you switch on the electric field in one direction and then you switch off and then you switch on in another direction. So, what will happen? The DNA will reorient first in one direction and move, and then as you switch off that and switch on in another direction, it has to move in another direction. So, this particular one exploits the length dependent reorientation time of DNA in electrophoresis, and the current is alternating between these two sets of electrodes at defined interval. So, here as separation is based on the ability of the DNA molecules to reorient and then move in the applied electric field. So, what will happen is that you have three kinds of, if we can just divide these large DNA fragments, and we are talking about DNA fragments which is bigger than 20 kb here. So, if I say small, it does not mean smaller DNA, but it is above 20 kb, but smaller in terms that it is smaller than say 100 kb or 1000 kb. So, there is smaller, medium sized and larger DNA molecules in that particular range. So, what will happen is when you switch on and switch off this electric field in different direction, then smaller DNA molecules will reorient faster and they will move also in the applied electric field direction. And as you switch off and then again switch on in another direction, they will move in another direction. They will reorient faster and then again move in another direction. So, they will be migrating little faster. Then there will be medium sized DNA molecules which will reorient, but they might not move too much as compared to the smaller size. So, they will reorient and move somewhat, but not equal to the smaller size DNA that is above a certain size. Then there are large DNA molecules which will spend most of their time in reorientation only, and they will not move at all. So, so you are able to, because of this disruption of raptation movement and exploitation of length dependent reorientation, different sized DNA molecules above 20 kb could be resolved in these gels. Now, here almost like million base pair DNA fragments, DNA molecules could be resolved by pulsed field gel electrophoresis. For example, many genomes like say of small organisms like yeast has been resolved in this technique. So, many lower eukaryotes which have chromosomes in say 0.5 to 3 MB size range could be resolved here. Pulsed field gel electrophoresis provides a means to characterize and map genes and chromosomes of these organisms. In higher eukaryotes, this pulsed field gel electrophoresis is used for long range restriction mapping with restriction enzymes that cut infrequently. So, a lot of applications are there for pulsed field gel electrophoresis. So, if I see this figure here, this kind of shows you that there are two DNA molecules, one is larger, another is smaller, and as one side in one direction current is applied, smaller ones moves little faster, and then as you switch on the electric field, then you can see the reorientation is taking place, and then a smaller one has migrated ahead of the larger molecule. So, this is how as you shift the field from one direction to another, smaller ones will reorient as well as they will move and migrate faster, and this is the basis of separation in pulsed field gel electrophoresis. Now, there are lot of variations of pulsed field gel electrophoresis, and we will see some of them here. Variations in this technique is based on changing the direction of migration, and the differences between the methods are primarily in the position of the electrodes. Remember the switching time which we were talking about, the time which is like for different direction applied electric field is called switching time. So, you switch on one and then to other. Mostly when you are switching in two directions, you make sure that the resultant vector is straight, and the whole and time and amount of electric field applied is such that they are moving in one direction only. Now, there are a lot of different kinds of pulsed field gel electrophoresis, which is like they could be moving back and forth in 180 directions. They could be like moving in at 120 degrees, or there could be a lot of other positions of electrodes in different directions. The clamp homogeneous electric field is probably the most popular method, and here in this particular technique, the apparatus are arranged in a hexagonal array, and there is a generation of homogeneous electric field. I will show you that how does it look like. The results in the lanes being straight and allows for an easy comparison of different samples, that is though there are so many directions, two directions, but you can load many samples at a time, and there are a lot of variations which could be tried. Now, different variations in pulsed field gel electrophoresis, there are a lot of variations. I have just listed 3 here, that is field inversion gel electrophoresis, where the field is applied field is switched on and off at 180 degrees, then clamped homogeneous electric field, where it is at 120 degrees, and there is a very advanced system called programmable autonomously controlled electrodes, and this could be you can control each of the electrodes separately and can generate lot of different kinds of variations. This is a very common way. This is a hexagonal array of electrodes you can see. Now, here each like this is shown, two directions are here shown. This is a gel placed in this electrode pattern, and the applied electric field is 120 degrees here, which is like plus 60 and minus 60 directions, and but resultant movement is in straight line in here. Now, let me show you this 3 variations which we were talking about on your screen here. Pulsed field gel electrophoresis, like I was telling you, this is a hexagonal array of electrodes, and which looks something like this. It is not perfect, but I think you can understand that. Now, you have like each leg contains this hexagonal array of electrode arrangement. Here each leg contains 4 electrodes. So, if I have to say I will just make this one, and these are 4 electrodes. These are electrodes are platinum, made of platinum wires, and so what you can do is, like for now the direction, the force on the DNA, if I say there is a DNA molecule here, if there is a gel placed in center of this electrode array and the samples are placed in here, then they will be in this direction, and another electrode when you switch on in second direction, that is, so this is, if I say from the center, this is plus 60 degree, and there could be another arrangement where you can have this in this direction, that is, I can make it here in. So, then these will be the negative electrodes, this could be switched on to the negative and this will be positive here. So, this is the clamped homogeneous gel electrophoresis, or you can call chaff. Clamped homogeneous pattern here is given, where you can change it to 120 degrees. There could be other patterns, like, so these 4 electrodes which are here in say field inversion gel electrophoresis, the pattern will be different. Alright, you have 6 lakhs in here, and in field inversion pattern, what you have is you have gel loaded in here, and you apply the electric field in 2 directions. One direction is in this direction, another direction is in this direction, that is at 180 degrees. So, accordingly these 4 electrodes which are placed in here will change their either they will be negative or they will be positive. They will be either negative electrodes or positive electrodes accordingly. Now, these are 4 of these are like I said, these are platinum wires. Now, remember, in field inversion gel electrophoresis, what you want is that the molecules should move in forward direction. So, always the time and the amount of electric field of the forward direction will be higher than the reverse direction. So, that the resultant movement is towards is forward movement, and that is done. In say program controllable pattern, each of these electrodes could be separately controlled. Like here, as we see it in clamped homogeneous or say field inversion, what was there, that each leg is controlled separately. But in case of field inversion, in case of programmable autonomous control system, you can control each of these electrodes is separately controlled, and you can generate various types of variations or directions in which applied electric field could be applied, and so it is much better. Now, if we compare them all these techniques, field inversion gel electrophoresis, you can separate up to 100 kb fragments or 100 kb DNA molecule. If you go to clamped homogeneous system, then where you apply plus 60 minus 60 directions or at 120 degrees to each other electric field is applied, there you can go up to say 2000 kb, and even the best results could be obtained up to 7000, it could be extended up to 7000 kb. And in programmable autonomous, you can have lot of different variations, and that way you can have much better results. So, this way these are 3 variations, there could be many more variations at different angles that could be found, where you can vary and you can get best results. Now, like I said, you can load many samples, and I have shown you, since the resultant movement is forward or in one direction, So, there is no problem, there will not be any mixing or so, and you can run lot of samples at a time. The factors which affect resolution are one is the uniformity of the two electric fields, that is very important, and you have to remember, like for example, in field inversion the forward electric field has to be higher and time also has to be higher. So, all these things has to be taken into account. So, homogeneous electric field has to be applied for best results. Lengths of the electric pulses is also important. So, that has to be seen, that has to be standardized. Then the ratio of the lengths of the pulses is important in and affects resolution. Then the angles of the two electric fields is important, and then the strength of the electric fields is important. So, all these factors has to be taken into account by while running pulsed field gel electrophoresis. Now, apart from electronics or the automation which has gone into pulsed field gel electrophoresis technique, where you can have lot of different directions and you can pulse the field as such, we can say. There is another important thing, when you are dealing with such big DNA molecules, these DNA molecules like say genomes and other, they can easily break, they are very fragile material, and in solutions there are shear forces which can break these into pieces, and then you will get smear, you will not get really good bands or good separation, and also your DNA material is broken here or it is disrupted. So, when you are dealing with very large DNA molecules, then DNA is not allowed to come into the solution. Rather, what is done is that DNA material is or the organism or the source from where DNA is being separated or isolated is directly, that whole thing is put into a plug which is low melting agarose or so, and so DNA is not allowed to come into the solution, and all the processing like say for example, deproteinization or breaking the cell wall, all those things will be done in that plug only, and the whole plug is then loaded onto the pulsed field in the slot, pulsed field gel electrophoresis in the gel, and so the DNA is not coming out into the solution, and the whole thing could be done very nicely. So, that is another important part of running pulsed field gel electrophoresis. All right, we come to the last part of this, that is RNA electrophoresis, which is also very important, but not as prevalent as DNA electrophoresis. So, like that of DNA, electrophoresis of RNA is also usually carried out in agarose gels, and the principle of the separation is based on size is the same. Often one requires a rapid method for checking the integrity of RNA immediately following extraction. So, RNA is highly susceptible to intra-strand H bonding, and secondary structures could be formed which will affect the migration through the agarose gel. So, what is done is you use denaturing agents or denaturing agarose gel electrophoresis is run, and certain denaturing agents are put in loading buffer. So, and RNA samples could be heated up to 65 degrees Celsius for 5 minutes prior to electrophoresis, and it could be run, and you can obtain the results in this form, where mostly what you see is two prominent rRNA bands are seen, which is 28S and 18S RNA, and then there is a slight smear of various sized mRNA molecules could be seen in here in the background. So, this is how the RNA electrophoresis can be done. So, this completes this lecture on electrophoresis of nucleic acids. Just to summarize, we have seen how DNA up to 20 kb could be run on agarose, simple agarose gel electrophoresis. Fragments above 20 kb cannot be separated or resolved on simple agarose gel, and pulsed field gel electrophoresis is the technique to be used for analysis, where the field is given in pulses in different directions, and where process of raptation is disrupted as we have discussed, and length dependent orientation of DNA molecule in electrophoresis is exploited. which can give you the separation of very large DNA molecules, even the genomes of small organisms like yeast can be performed there. And then we have discussed about a little bit about RNA electrophoresis. So, this completes our chapter on nucleic acid separation on electrophoresis, and in the next lecture we are going to discuss about the immunoelectrophoresis and capillary electrophoresis. Thank you.